If no other questions, I'm going to go ahead and move on with the next paper, which is coming to us from some of our international submissions here outside the United States and including the United States, which is kind of fun. So I'm going to read off all the institutes that are involved in this trial, which is quite extensive. We have investigators from the Karolinska Institute, ITMO University in St. Petersburg, Stanford University, Royal Institute of Technology in Sweden, and the Karolinska University Hospital and Leiden University. This is somewhat of an interesting paper, and similar to the last one, it's a lot of good descriptive data that is really going to prompt a lot of future research in this field. And Dr. Matic is going to present it for us, and maybe you could pronounce your first name so I don't butcher it for you.
Yeah, thank you for the introduction. My name is Ljubica Matic, and indeed, this is how we like to work. In many of our papers, we have had a lot of collaborators. We like to work in an international way, and you'll see I will get back to that by the end of my presentation. So let me quickly share screen by just giving you a snapshot with the visual abstract. You can have a look at it for 2 or 3 seconds until I put up my presentation. And then I will go full screen with the presentation. So I hope you all see my slides now. So I'm very, very happy to be invited to talk in this forum. As the editor said, this is one of the studies published in the inaugural issue. We are very honored to be part of this inaugural issue, of course. And the rationale for our study was the realization that there is still a need for better understanding of molecular mechanisms underlying vascular injury and healing. So we know that endovascular interventions can cause arterial injury. They can induce a healing response. However, this healing response may lead to complications of either defective or excessive response. These complications are common and may result in repeated interventions. And we think that in this field there exist well-established experimental models of intimal hyperplasia that can still be used for understanding the underlying mechanisms and eventually helping to resolve the clinical problems. So the aim of the study was very simple, in fact, to systematically investigate the reaction to vascular injury over a prolonged period of time, however, using new available methodologies. And here I really would like to mention that we have generated a large resource for the purpose of this study. I am showing you here in the corner a picture of a Swedish Viking settlement, which is called Birka, the same as the name of our biobank, the Biobank of Rat Carotid Artery Injury. And I also want to mention that this will be a tribute to Professor Alec Kloves, who invented this method, in fact. So we started by performing balloon injury of the carotid artery in about 70 Sprague Dawley rats. We did that over a long period of time and systematically collected the samples, as you can see here, over 12 weeks. We collected both blood and tissues from different organs, including carotid arteries. And we isolated also RNA from these samples and did global microarray profiling to analyze global gene expression differences and pathways in response to vascular injury. So what we have seen very simply is that over a 12-week period, there is a very distinct pattern of gene expression changes that appear in the injured artery. So there is an early response within the first 2 to 20 hours after injury. There is also an intermediate response. And there is something that we call the late phase towards the resolution. However, these distinct gene expression patterns one cannot observe in the uninjured contralateral arteries although there are gene expression changes in the uninjured artery as well, so it should not be considered that no changes are happening there. We next looked at the pathways, trying to understand these large lists of differentially regulated genes at each time point. And we could see that in the early phase, there is primarily immune activation and coagulation going on. In the intermediate phase, pathways were very dynamic, especially at 20 hours and at 5 days after injury. And pathways could be attributed to primarily smooth muscle cell modulation, proliferation, migration, and extracellular matrix remodeling. And finally, in the late stage, we could see that there was an attenuation of these pathways, an attenuation of the gene expression changes in general, and reacquisition of contractile features, at least on the gene expression level. This is the notion that we got. Using these technologies, we could even assess some novel pathways that were previously not reported in this model. We combined here transcriptomic analysis with immunohistochemistry in the arterial tissue to localize the genes and validate the changes on the protein level. And we could see, interestingly, that there was a very early induction of the stem cell markers in this tissue, exemplified here by OCT4. And these stem cell markers were first identifiable in the vasculogenic zone between the adventitia and media, but then from 2 weeks on, we could actually localize these signals to neointimal smooth muscle cells. Next, we noticed also an early induction of the immune cell markers. However, these markers could not only be attributed to inflammatory cells, as one could expect, but exemplified here with the macrophage marker, they were actually localized to neointimal smooth muscle cells also at 2 weeks after injury. And finally, we could even detect a little bit later on an induction of osteochondrogenic markers, exemplified by transcription factor RUNX2 here, that also could be localized to neointimal smooth muscle cells. So really a very complex interplay of novel pathways that should also be investigated in this model. I mentioned here osteochondrogenic markers, even though there's actually no visible calcification in the model. So in conclusion, vascular injury induces transcriptional landscape and also metabolic changes, which is something that we looked at in the paper, identifiable as early, intermediate, and late response phases, reaching homeostasis after several weeks. Our study provides a temporal roadmap of vascular healing that we think now can be used for exploration of novel pathways and also to come up with novel therapeutic targets to treat vascular complications. And our key message here, you probably notice I have simplified the scientific part of this paper very much. You can, of course, go and read the full version if you are interested. But the key message for the community is that we have generated here what we think is a very valuable resource, a resource that contains a database with transcriptomic longitudinal tissue profiles, and also a biomaterial repository that we would like to share in collaboration with the other interested parties in this research community. So please feel free to contact us. Finally, I want to thank a very large vascular surgery unit at Karolinska. We have an amazing tradition of almost 50 years. We are led by Professor Ulf Hedin, who is also one of the editors in this journal. And we had, as it was already mentioned, a fantastic alignment of collaborators from Russia, our long-standing collaborators from Stanford, Professor Thomas Pertermos, and also from Leiden, from the Netherlands. And I especially need to thank the team who worked on this particular study. So our PhD students, Ula, Bianca, Till, and Samuel, our technicians, Mete and Malin, and finally, senior researcher Anton Razuvaev and also Ulf Hedin, who together actually conceived and designed this whole study. And maybe it's of importance to mention that the biobank and the injury model in rats, it was done by professional vascular surgeons. The rest of us here in this picture are basic researchers. We didn't do the surgery. Thank you so much.
Absolutely fantastic. Thank you so much. All right, let's jump into the questions, because this is a lot to be unpacked. We have the first question here is very nicely presented an important systematic approach to neointimal hyperplasia. Can you comment on differences between neointimal and adventitial signals?
Yes, well, an interesting question, of course. We have not particularly looked at the adventitial changes. This is a part of the tissue that was not part of the microarray analysis, so I don't think I can comment on that. It also was not specifically assessed in the histological analysis. That would probably be possible. So I don't want to give any more comments, but if there is anybody who is interested in this and if we can help anything, feel free to contact us. We are in the mood for collaboration.
Excellent. And if somebody was interested in leveraging the biobank, is that something they do through you, or do they contact somebody else just at Karolinska? How would they go about making that connection? Well,
the community maybe knows us. We are, in that sense, we are profiled very much when it comes to biobanking. We like to apply human biobanking philosophy to animal models and even to cell culture. We think that, especially when it comes to animals, anything you do should be done large scale and with the possibility that many people can analyze it without the need to actually repeat a model which is difficult for all of us to learn, if not impossible. So we will have, or we have, a little steering board that serves just for the purpose of comparing these collaborative requests so that there is no conflict of interest. But they can feel free to contact Anton Razuvaev, myself, or Ulf Hedin, and we will very quickly respond with an answer. In principle, there's really very little overlapping interests. Our own interest is in smooth muscle cells, so that's all I can declare right now.
Excellent. Thank you so much.
Thank you.
All right. We had some great questions.